course title Microbial Technology. The video lecture title is Technique of Pure Culture, and the lecture number is 16 and under Chhattisgarh Swami Vivekanand Technical University, Durg. In previous topic, we already discussed about the measurement of growth and growth yield. A few bacteria are inoculated into a liquid growth medium or any solid medium. The population is counted at interval. Some of the standard method of measuring microbial growth are as follows, like direct microscopic count, quantification via electronic means, the plate counting technique, bacterial number estimation based on turbidity, nitrogen content determination, cell dry weight determination, etc. Bacterial factor affecting bacterial growth, concentration on nutrition, temperature, concentration of gas, pH, ion concentration and salt concentration, and the availability of water. These are some factors which affecting the bacterial growth, which we already discussed. Now, in current topic we are covered the topic pure culture technique, about that introduction of pure culture technique, importance of pure culture technique, its history, pure culture techniques under that sterilization, inoculation, isolation, then isolation techniques, a special method of isolation, culture media and types, purity of culture, maintenance and preservation of pure culture by short term preservation and long term preservation method, and its summary. Now, introduction. Pure culture technique. What is culture? Culture act of cultivation of microorganism. Mixed culture is the more than one type of microorganism, and the pure culture is containing a single species of organism. Typically, a pure culture is created by the diluting the sample several times prior to inoculating the freshly prepared medium, or by transferring a small amount into a fresh sterile growth medium and spreading the individual cell evenly throughout the medium surface. Importance of the pure culture technique. Its importance in microbiological studies, find out the desired microorganism. It is useful to identify correct microorganisms for accurate studying and diagnosed disease in field of medical science. Testing, experimenting with a pure culture ensure that the same result can be achieved regardless of how many times the test is repeated. Pure culture spontaneous mutation rate is low and the pure culture clone is approximately 99.99% identical. History. Louis Pasteur during 1822 to 1895 and the Robert Koch during 1843 to 1912. Both are known recognized person in the field of microbiology. Louis Pasteur is the father of microbiology and the Robert Koch is the father of practical bacteriology. In 19th century lead to several keys breakthrough in medicines. Agar was discovered around the 1658 by the Minoya Terajomon in Japan. Agar was first used in microbiology in 1882 by the German microbiologist Walter Hayes as assisting work in Robert Koch laboratory. Pure culture techniques consist of three interrelated techniques. First is sterilization, second is inoculation, third is isolation of single cells or microbial species. Sterilization, it is the total elimination of all other microorganisms from the exterior or culture medium. They are categorized in the two category. First one is the physical method and second one is the chemical methods. In physical method, we know three main method: by the heat, by the radiation or by the filtration. In heat, we use both type of heat, dry heat and moist heat, which we see in that particular table or chart. Red heat, flaming, incineration, hot air oven are the dry heat method, and during the moist heat we use below the 100 degrees Celsius temperature, at 100 degrees Celsius temperature and the above the 100 degrees Celsius temperature with moist heat. Then in case of radiation, we use ionization radiation like infrared or ultraviolet, or ionizing radiation like X-ray and gamma rays. In filtration, we use three different type of filter. First one is depth filter, second one is membrane filter, and third one is air filter. This is the physical method of sterilization. This is the figure of the physical method of sterilization: incineration, red heat, flaming, tyndallization, boiling, autoclave, hot air oven, pasteurization, radiation, and filtration. The chemical method is also divided into the two parts, liquid and gaseous. In liquid part, we use alcohol, aldehydes, phenolics, halogens, heavy metals, surface active agents and dyes, etc. for the sterilization purpose. And the gaseous method, we use formaldehyde, ethylene oxide and beta propiolactone as a gaseous chemical for the chemical method of sterilization. By both means, we sterilize the equipment and glasswares. Now, isolation of pure culture. First of all, inoculation. Its basic techniques to place microbial culture onto a culture medium. It is performed using an apparatus called inoculation loop made up of platinum or nichrome wire. Obtaining pure culture or isolation of pure culture. Culture composed of cell arising from a single progenitor. What is progenitor? Progenitor is termed as CFU. Then aseptic technique prevents contamination of sterilization of object. Then the common isolation techniques are four: strict plate method, split plate method, serial dilution method and pour plate method. Now, strict plate method. Strict plate method is based on principle of dilution. It can be described as a rapid qualitative isolation technique. The main creation of isolation is to obtain a reduced number of colonies. In this technique, a loop full of culture is spread on an agar plate to get individual cells far apart enough from each other. The streaking method gradually dilute the inoculum such that the bacterial cells can be counted as colony forming units. Type of streak plate method: quadrant streaking. This is the most common method of streaking, where the petri plate is divided into 4 quadrants and then inoculated. It is also known as 4 quadrant streaking. A loop pull of the inoculum is taken and streaking such that the first quadrant contains the highest concentration of the inoculum, following by the second quadrant, third quadrant and fourth quadrant. This is a discontinuous method where the loop is sterilized after streaking in every quadrant. By the time the fourth quadrant is streaked, the inoculum is diluted enough to give rise to individual colonies. The T streaking. In this method, the petri plate is divided into 3 quadrants by drawing a letter T. Similar to the 4 quadrant streaking, it is discontinuous method where each quadrant is streaked after loop sterilization and quadrant that is streaked last gives isolated colonies. Continuous streaking. In this type of streaking, the inoculum is spread from one edge to the center of the plate. The plate is rotated 180 degrees Celsius and the remaining portion is streaked without sterilizing the loop. Another form of continuous streaking is used for this diagnosis purpose. The petri dish is divided into sections and different cultures such as urine, sputum, pus, etc. are the streaked in each section to get maximum output. Radiant streaking. In this streaking method, the inoculum is spread one on edge of the petri dish from the edge. Vertical lines are streaked in the upward direction. Next, the vertical lines are streaked horizontal to obtain pure isolated cultures. Advantage. This is one of the most popular technique used to obtain isolated colonies of bacteria. It finds a great application in biotechnology as it can be used to identify transformed bacteria from non-transformed bacteria by adding an antibiotic to growth medium. It also finds great application in diagnosis purpose. This is the figure in which we show all types of streaking method. Next, pour plate method. The pour plate method is a plating technique that is commonly used to obligate an anaerobic bacteria. This technique is used to isolate microbial colonies by serially dilution and then counting the colony forming unit, in short CFUs. In this method, the liquid sample is poured into a Petri plate before the solidification of the agar medium. After solidification, colonies grow both inside and on the surface of the medium. However, the colonies growing inside the medium are confluent, those on the surface are used for the viable counting. The optimum count of the colonies obtained must be between 20 to 300 CFU per ml. If it is more or less than this range, the whole process needs to be repeated. Advantage: it is useful for counting viable colonies. It can detect very low loads of bacterial count as well. It does not require previously solidified agar plates. It can also be used for clinical and environmental samples. This is the figure of the pour plate method. Now, split plate method. The split plate method is microbiological laboratory technique for isolating and counting the viable microorganism present in a liquid sample by spreading a certain volume of the sample over an appropriate solidified culture medium. Following the incubation in a successful split plate, there will be the formation of the evenly distributed discrete colonies all over the surface of the culture medium. The technique is used for the isolating and counting the total number of the viable microorganism, that is calculating the colony forming units per ml in the given sample. Advantage is this is simple, easy and quick method of the culturing microorganism. A very low microbial load can be detected. The colony morphology of the microorganism can be studied by this method. The size of the colonies produced when cultured by the method will be larger than those produced by the sample species using the pour plate method. It is qualitatively as well as quantitatively isolation method which facilitating isolation as well as enumeration, that is calculation of CFU per ml of the microorganism. It can be used in performing Bohr antimicrobial activity testing. Any clinical or industrial or environmental sample can be used unless it is in a liquid state or can be dissolved to prepare a suspension. It is used in preparing the maintaining stock culture. It is most appropriate method for the culturing the aerobic microorganism. Even the syntrophic bacteria can be isolated because they both can be grown in a single plate nearby these distinct colonies of each type. There is very least chance of contamination if one used sterile glass bead as a spreader. This is because all the action will be done by closing the lid of the culture plate. This is the figure of split plate method. Now, serial dilution. Serial dilution, as the name suggests, this is a series of sequential dilution that are performed to convert a dense solution into a more usable concentration. In simple words, serial dilution is the process of stepwise dilution of the solution with an associated dilution factor. In biology, serial dilution is often associated with reducing the concentration of the cells in a culture. To simplify the operation, the following is the procedure for a 10-fold dilution of a sample to a dilution factor of 10 to the power 6. The sample is taken in the test tube and 6 test tube each with 9 ml of sterile diluent, which can be either distilled water or N saline, are taken. A sterile pipette is taken. 1 ml of properly mixed sample or culture is drawn into the pipette. The sample is then added to the first tube to make the total volume of 10 ml. This provides an initial dilution of 10 to the power minus 1. The dilution is thoroughly mixed by the empty and filling the pipette several times. The pipette tip is discarded and the new pipette tip is attached to the pipette. Now, 1 ml of the mixture is taken from the 10 to the power minus 1 dilution and is imparted into the second tube. The second tube now has total dilution factor of the 10 to the power minus 2. The same process is then repeated, remaining tube taken 1 ml from the previously and adding into the next 9 ml diluent as the 6 tube are used. The final dilution of the bacterial per cell is equal to 10 to the power minus 6. This is the figure of serial dilution in different test tube, and what we find in the last tube, it is microbial counting is very low. Now, a special method of the inoculation of pure culture. First is single cell in isolation method and second one is enrichment culture method. In single cell inoculation method, we use basically capillary pipette method or micro manipulator method. In enrichment culture method, we use a specific enrichment media for designed for the specific purpose and environment by the incorporating a specific nutrient in the medium and by modifying the physical conditions of the incubation. The culture media is gel or liquid then that contain nutrients and is used to grow bacteria or microorganisms. They are also termed growth media. There are two types. First, defined media, second is complex media. Defined media are prepared by the adding a strict composition of the highly purified inorganic or organic chemicals to distilled water, that is NAM, nutrient agar media. And this is the composition of nutrient agar media. Now, the complex media are prepared using digested of microbial, animal and plant products like casein, milk protein, beef extract, soya beans, tipped soya broth and yeast cell for the yeast extract media. Based on the consistency, media are divided into three types: solid medium, semi-solid media and liquid or broth media. Example of the solid media is McConkey agar media, chocolate media, nutrient agar media. In all media, we contain agar for the solidification purpose. And the semi-solid media, it is the media in which base is the mid condition, means 50-50 solid and 50% is it is liquid condition. Example of semi-solid media is motility test medium and Ames test transport medium, etc. Liquid broth media. Example of liquid broth media is nutrient broth media, glucose broth media, alkaline peptone water media, etc. In this media, agar is not used and this is in liquid condition. Based on their practical use, media are divided into 6 types. First is general purpose media, example peptone water, nutrient broth, etc. Enrichment media use specific enrichment nutrients such as blood agar media, etc. Selective media or enrichment media, that means mannitol salt agar media, that is specific for the high salt concentrated growing microorganism. And the differential or indicator media, it is contain specific indicator for the differentiated between the two types of microorganism. Transport media, it is a media used for the streptococci from throat specimen. The anaerobic media, it is specifically used for the Clostridium species. Assay media, it is used for the antibiotic assay media. In minimal media, a media that contains only inorganic salt, a simple carbon source, etc. Now, minimal media is the media which contain very less amount of a specific nutrient. This is the figure we show agar plate and the step culture, broth culture, slant culture, etc. Now, proof of purity of culture. A pure culture is one in which the cell are all of one kinds, that means each are look like evenly and the microorganism look alike microscopically and strain in the same fashion. When plate, all the colonies formed look alike. Stick, stab, etc. are uniform. Several isolated colonies perform identically, that is for same. Now, maintenance and preservation of pure culture. After obtaining the particular microorganism as a pure culture, it is essential to preserve the microorganism viability and purity by preventing contamination of the pure culture, since repeatedly subculturing is the time consuming. It becomes difficult to maintain a large number of pure culture successfully for a long time. There are two strategies for keeping culture alive. First is slant culture, second one is step culture. Objective of the preservation. The primary aim of the preservation is to maintain the organism for extended period in a viable condition. Second, to keep the culture isolates uncontaminated for a longer period to be used for educational research purpose. Then variation or mutation, that is to preserve the culture in condition, that is original, maintain as a original isolate. Preservation method. There are two ways by which keeping culture preserved: short term preservation method, long term preservation method. Frequency of transfer, immersion in oil, freezing at -20°C temperature, drying and storage in distilled water are the short term preservation method, whereas using liquid paraffin and mineral oil, using of glycerol, immersion in distilled water, using sterile soil, lyophilization and cryopreservation, these are long term preservation method. Short term culture preservation. Normally in laboratory, the pure culture are transferred periodically onto or into a fresh medium, subculturing to allow continuous growth and viability of microorganism. Microbial culture remain viable for several weeks or months on a medium like nutrient agar. This is simple method and any simple apparatus are not required. How it is easy to recover the culture. The transfer has the disadvantage of failing of prevent changes in the characterization of a strain due to the development of variants and mutants, and risk of contamination is also more in this process. Short term preservation method, periodically transfer a fresh media. Strain can be maintained by periodically prepare a fresh culture from the previous stock culture. The culture medium, the storage temperature and the time interval at which the transfer are made vary with the species and must be maintained beforehand. Next is storage in the saline suspension. Bacterial culture is preserved in first salt concentration in screw caps and tubes to prevent evaporation. The tubes are stored in room temperature. Whenever needs, the transfer is made on agar slant. Third is preservation by drying method. So, spores of some microbes which are sensitive to freezing, drying can be prevented by drying from the liquid state rather the freezing state. Different procedure of drying method are follows. First one is paper disc method, second one is gelatin disc, third one is L drying method. Now, preservation by the refrigeration. Pure culture can be successfully stored at 0 to 4 degrees Celsius temperature either in refrigerator or in cold rooms. The method is applied for the short duration 2 to 3 weeks for bacteria and 3 to 4 months for fungi, as the metabolically activity of the microorganism are greatly slow down, but not stop. Their growth continuously slowly as nutrients are utilized and waste product released into the medium. Finally, result is the death of the microbes. Long term preservation. Long term preservation methods permit interval of the months or even years between the subculture. Isolates may be stored identify indefinitely if they are maintained frozen at minus 70 degrees Celsius temperature or below this. Temperature can be achieved in an ultra slow freezing minus 70 degrees Celsius temperature or liquid nitrogen freezers minus 196 degrees Celsius temperature. Lyophilization or storage at minus 70 degrees Celsius temperature or below is the best method for the long term preservation of bacteria. Culture and general storage of the isolates at minus 20 degrees Celsius is not recommended. Long term preservation method, preservation by the using paraffin. This is a simple and most economical method of maintaining pure culture of bacteria and fungi. The layer of paraffin ensure anaerobic condition and prevent dehydration of the medium. This condition help microorganism or pure culture to remain in dormant state and therefore, the culture can be preserved for months to years. Preservation by glycerol. Bacteria can be frozen using 15 degrees Celsius 15% of glycerol. The glycerol is diluted at 30% and the equal amount of glycerol and culture broth are mixed dependent into tubes and frozen at minus 10 degrees Celsius temperature. After mixing our sample, freezing method at minus 80 degrees Celsius to ensure that they remain viable for as long as possible. They can stored at minus 20 degrees Celsius temperature and transferred after 12 to 18 months. Immersion in distilled water. This is one of the method in expensive and low maintenance method for maintaining fungal culture. So, to immerse them distilled water, fungi can be stored in this method at minus 20 degrees Celsius for up to 10 to 20 years depending upon the species. Preservation using sterile soil. It is mainly applied for the preservation of the sporulating microorganisms such as Fusarium, Penicillium, Alternaria and Rhizopus etc. Soil storage inoculation of 1 ml of spore suspension into soil. Autoclave twice and incubating at room temperature for 5 to 10 days. The initial growth period allows the fungus to use the available moisture and gradually to become dormant. The bottle are the stored in refrigerator and the microorganism can be found viable after 70 to 80 years. Lyophilization method, it is a vacuum sublimation to that involve freezing drying. The culture using products that are hygroscopic protect them from moisture during storage. Using vacuum sublimation, the cells can be effectively preserved. Freezing must be very rapidly with the temperature lowered to well below 0 degree Celsius temperature, as such minus 20 degree Celsius temperature. Lyophilized culture are stored in the dark at 4 degrees Celsius in refrigerators. Many microbes preserved by method can be remain viable and unchanged in their characteristic for more than 20 years. Advantage: only minimal space is required and hundred of lyophilized culture can be stored in a small area. Sterility can be maintained for a long period of time. This is the graph by which we can see the time and temperature of the lyophilized microbes. Now, disadvantage: handling and processing time increase sterile diluent. Sterile diluent is needed upon reconsistency. Then equipment become costly and complex. Out all tools can be freeze dried. Slow process average cycle is 42 to 47 hours and high energy cost. Now, cryopreservation method. As we know about the cryopreservation, it is a liquid nitrogen method at minus 196 degrees Celsius temperature. Cryopreservation involves freezing culture in liquid nitrogen at minus 196 degrees Celsius or in the gas phase above the liquid nitrogen at minus 150 degrees Celsius. That helps in survival for long storage time. In this method, the microorganisms are rapidly frozen in liquid nitrogen at minus 196 degrees Celsius temperature in the presence of cryoprotectants such as glycerol or DMSO that prevent the cell damage due to the formation of ice crystal and promote cell survival. This liquid nitrogen method has been successful with the species that cannot preserve by lyophilization and the most species that can remain viable under these conditions for 10 to 30 years without undergoing change in their characteristic. Advantage: the culture do not undergo genetic mutation, therefore maintaining the original characteristic. Disadvantage: it is a expensive method, reduce risk of contamination. Culture can be preserved for long period. These are some references. Topic covered in the next lecture is cellular respiration is a process that takes place inside the cell where energy is released by the breakdown of glucose molecule. The process can be convertedly divided into two categories based on the uses of oxygen, namely aerobically and anaerobically respiration. The primary difference between the aerobic and anaerobic respiration is the presence and absence of the oxygen during the process. Aerobic respiration can be found in the cytoplasm and the mitochondria, whereas the anaerobic respiration found only in the cytoplasm. All higher organisms such as mammals have aerobic types of respiration, whereas anaerobic respiration occur in the lower organisms such as bacteria, yeast, etc. Byproduct of the aerobic process produce carbon dioxide along with the ATP, the energy currency of the cells, whereas byproduct of the anaerobic respiration process are lactic acid and ATP. Thank you.